Hey guys, Mrs. Spurlock here. We're going to start this week by talking a little bit about office hours and my expectations. My Zoom account is having some security issues, so for the remainder of the year, if you plan to attend office hours, you will need to let me know in advance, and I will invite you to those meetings so that I'm not putting the meeting ID anywhere on the internet that other people can use it and get into the meeting when they're not invited. All right. Now, chapter 5, lesson 4 is about human inheritance patterns. Now, a lot of this chapter is going to be things that you guys are already familiar with, that you've already been doing, because we talk a lot about human inheritance while we're doing other forms of inheritance, okay? All right, so chapter 5, lesson 4. Go ahead and share my screen here. Chapter 5, lesson 4 is about human inheritance, like I said before. And we are going to have two big questions this week. Number 1, what are some patterns of human inheritance? And number 2, what are the functions of the human sex chromosomes? Feel free to go ahead and pause and copy this down, or this PowerPoint will be available via Google Slides. It will be attached to the same assignment that your lecture is attached to. What are some patterns of human inheritance? Big question number 1. The first pattern of human inheritance involves a single gene with 2 alleles. These are simple Mendelian traits. These are like the pea plants with purple versus white flowers. In humans, dimpled chin is an example of a single gene with 2 alleles. You either have it or you don't. Single gene means that there's only one type of letter. Okay, so when we're talking about alleles and we use letters to represent them, if there's a single gene, there's only one type of letter. So for dimpled chin, I'm going to use letter D. And I can either have a capital D or a lowercase d. So let's say I've got a homozygous dominant individual, and I'm going to cross them with a homozygous recessive individual. Okay, it would be just a really simple Punnett square. If you were doing a Punnett square for a single gene with only 2 alleles, it's the most simple form of Punnett square that we would typically do. In a single gene with 2 alleles, you only have 2 phenotypes. So for a dimpled chin, you either have a dimpled chin or you don't have a dimpled chin. And a reminder for anyone who maybe has been relaxing a little too hard and doesn't remember what phenotype means, remember that phenotype means physical appearance. So for a single gene with multiple alleles, this is going to be the exact same type of inheritance that we've learned about with multiple alleles already. And our example for a single gene with multiple alleles is blood type. Okay, so right here is where you need to be paying close attention, please. Now, for my phenotypes in blood type, they're going to be A, B, or O. Genotype is going to be a little different. Because I have multiple alleles, I can't just use A, B, and O because of the way the dominance patterns work out. Right, so we use the letter I with a superscript, so either A or I, B, and lowercase I. And the reason we do that is because A and B can be co-dominant. The reason we use superscripts is because both A and B are dominant over lowercase I, but they're also co-dominant. So if I have type A blood, I either have capital I, capital A, capital I, capital A, or that would be homozygous for type A blood. I can also be heterozygous for type A blood, where I have a capital I, capital A, and a lowercase I. And type B blood follows this exact same pattern. So if I'm homozygous for type B blood, I'm going to have 2 capital I's with the capital B's, or I can be heterozygous for type B blood where I have a capital I, capital B, and a lowercase I. Type AB blood is where we see the co-dominance of the A and the B. Type AB blood can only have one genotype, and that's capital I, capital A, capital I, capital B. That's the only way to get type AB blood. And then the only way to get type O blood is to be homozygous recessive. The next inheritance pattern in humans that we're going to talk about is multiple genes. These are also called polygenic. We've talked a little bit in class about polygenic traits and how they fall on a large spectrum of phenotypes. So my favorite example, because I think it's the easiest to understand, is height. Okay, and so when we're talking about phenotype, there are so many different heights of all of the students. If we have 170 students, we could have 160 different phenotypes. We could have 160 different heights depending on how precisely we're measuring. And that is it for big question number 1. The type of inheritance patterns in humans that we talked about are a single gene with 2 alleles, a single gene with multiple alleles, and multiple genes. Does that mean those are the only ones? Definitely not. There are many other forms of inheritance patterns, but those are the 3 that we are going to be talking about. All right, so big question number 2 says, what are the functions of the sex chromosomes? So if you recall, when we were talking about human chromosomes, we talked about how there are 23 pairs of chromosomes, meaning a total of 46 individual chromosomes. Of those 46 chromosomes, 2 of them are sex chromosomes. If a person is a female, then those 2 sex chromosomes are both called X chromosomes. And X chromosomes are a little bit larger than Y chromosomes. And if a person is male, one of those sex chromosomes is an X and the other one is a Y. And that's what makes them biologically or genetically, that's what makes someone male versus female. So if we wanted to talk about the way sex chromosomes determine if a fetus develops into a male or a female, the first thing we have to talk about are the sex chromosomes of the parents. So like I just mentioned, in order to genetically be a female, a person has 2 X chromosomes. So the genotype of a biological mom is going to be 2 X chromosomes, while the genotype of a biological father has to be an X and a Y. Now, what we can do next is we can make a Punnett square, and we can figure out the likelihood that someone is going to have a boy versus a girl. So if a woman and a man that are genetically have the right number of sex chromosomes, if they have offspring together, we can put 2 X's along the top and an X and a Y along the side, or vice versa, it doesn't really matter. And what you're going to see is that genetically, when it comes to our chromosomes, there should always be a 50% chance that a fetus is going to be male and a 50% chance that a fetus is going to be female. And every year when we talk about this, people say, oh my goodness, well, I have 7 brothers and 1 sister. And that is because the gender of the first child has no effect whatsoever on the likelihood of the gender of the next child. So every single time it's a 50% chance and it never changes. Sometimes genes are found on sex chromosomes and we call these sex-linked genes. One type of gene that we know is sex-linked is the gene for color vision versus color blindness. We know that it's found on the X chromosome. So an X chromosome with a big C is normal color vision, and it's dominant. Meanwhile, if we have X lowercase c, and again, I've talked a little bit about how when you're doing a Punnett square, try to use letters that look different as a capital and a lowercase. C is not one of them, but it's the example in your book, so I'm going to use C for now. X lowercase c is going to code for color blindness. Another thing that we see with sex-linked genes is that recessive forms of sex-linked genes happen significantly more often in men than in women. And we're going to talk about why that is right now. So if we have X, capital C, X, lowercase c, I'm just going to make it little itty bitty. So this is a woman who has normal color vision, but carries the gene for color blindness. Let me make that a little bit nicer. I'm going to make it bigger too. And this woman is going to have some kids with a male who has normal color vision. So if we put these letters into our Punnett square, what we're going to see is X capital C, X capital C. In this box, this tells us there's a 25% chance that these two people will have a daughter, and she's going to have normal color vision. In the next box, we would get X capital C, X lowercase c. This tells us that there's a 25% chance they'll have a daughter that carries color blindness. That means that she herself can see normal color vision, but she is able to pass on the trait for color blindness. All right, and in the next row, we're going to get X capital C Y. This is a son with normal color vision. And the last example is going to give us an X lowercase cy. X lowercase cy tells us that there's a 25% chance that any of their offspring could be a colorblind male. This should show you why more males end up developing things like color blindness than females. Because females have two X chromosomes, they have an opportunity to cover up color blindness. So a female can be carrying color blindness but not have color blindness. Meanwhile, a male only has one X chromosome, so if they have the gene for color blindness, then they can only be colorblind. All right, and that is going to do it for both of our big questions today. If you have any questions about the packet, please either email me or you can come to office hours. I need to make a request of everyone. Please, no more socializing in the Google Classroom. You may reply to other people's discussion board posts, but there needs to be no more comments where you guys are just chit-chatting or asking each other how you're doing. That needs to be done on a different platform because I am missing important questions from people because I'm getting flooded with notifications that you guys are talking about, I don't know, whatever it is that you guys talk about. So please, no more socializing on Google Classroom. Okay, bye.